Oh, okay.
Still 5 minutes. So. And then we can get the next come in.
So welcome to the afternoon session B, on how forest genetic resources can be used to improve population adaptation to climate change and boost the adaptive potential and both for manage and natural populations. And so we have 4 regular talks and 4 presentations, and then the keynote at the end, Tongli Wang at distance. And so we will have 3 regular talks of 16 minutes and then questions, and then the break, and then after 1 regular talk and the flash presentations. At the end of that, the questions for all, and then the keynote. So okay, that's it. And
hello, everyone. I will present the future climate effects affected the Norway spruce in the future. How the climate change affect spruce, because the spruce is grows in the whole Europe. This study was based on the one of the biggest Provenance Trials. So the aim of this project, this study based on the B4EST project, we wanted to build one of the universal function to predict the ones of Norway spruce to the future climate, and we will interpret the provenance effects and also the genetic and the environment effects for each provenance, and then to predict the population and also the species, how they can be shifted range-wide for the future climate, and guide the assisted seeds in the future. Based on the UFRO, we have the UFRO built 3 big provenance trials. This one is the second time. Previous in 1930s, there is one like UFRO provenance experiment. This one is the second time. Then they started to connect as many as possible the provenance. So more than 1,100 provenance was collected from the whole range-wide. But many is focused on the outer area. Outer area since has too many. Then late at the beginning of the designs, these 1,100 provenance was divided into the 11 groups based on, so each group, each block actually is 100 provenance, but they covered the similar areas. So each block 100 provenance could be, actually they are cover whole range wide, so it's more or less is similar. So we can consider they are genetically more or less similar. And then later the Swedish Peter Kluge, he defined this 1100 provenance into 90 groups based on 96 groups based on the geographic information and also the blood those information. Then later this 1100 population or provenance is further defined to 20 groups, but the remaining of the about 20 provenance, which I defined because they are as the 21 groups, because they are located in the Russia. Originally, the tallest scope, he did not group this Russia population, South Russia population. Then these 1100 provenance was originally should be located for 20 trials, but each trial could be located in several sites. For example, the Swedish one, this top 3 trials, they are located in the same place. So there is one trial is one site, but for the German, one of the German trial, they are located in nine locations, so 9 sites. Originally, the UFlow was planning to run in 20 trials. Each trial include all the provenance and 8 or 25 individual for each provenance, means in the same trial. Actually, later, no one get 2 trials. No one get data for the England trial and also Finnish trials. So there is no phenotypic data available. Based on this project, we started to contact all the groups, all the participants in 1918. So just we did not get one of the trials, but the rest is okay. We finally get it. Most of the trades was measured by many years. Usually we measure height and DBH, also sometimes survival and the wood quality trait, even in one of the trials in France. So in this I just use the height. Most of the trials they measured in 11 years old. But some of the trials, like the T19 is in Polish trial, they are located in 2 places, but they did not measure in 11 years old. And also the Hungary trial, they measured in different, 9 years old in 1972 and then 1977. So the data is very heterogeneous. So I did several steps. First, I need to adjust the age effect as long as they are not measured in 1973, and also adjust the block effect, also filtering the redundancy of the provenance. Because the too many provenance in the outer area, also the Central Europe. Here is the example how to adjust the age effects. If there is several age of the measurements, then without the age 11, we could use the polynomial function. Actually, is very accurate as long as they are provenance mean, because we are using each dot is the provenance mean. So those models without measurement for specific year will be very accurate. Most of the measurement is for this UFlow trial. For height is usually less than 20 years old. So at the beginning is grows slowly, later grows fast. Step 2 is just the block adjustment. It's very just fix the effects for the block. But step 3 is just we need to filter the those place is too many nodes compared to the northern Scandinavia area. But in the northern Scandinavia, since we don't have the provenance even, so I filtered like more than about 700. So finally, we just use less than 500 provenance for the modeling. And also I use the Climatic DT to extract the climate data for site and the provenance. For the modeling, we just use the individual response functions for the each provenance. We wanted to see how they respond for the climate variable for the site for each provenance. So we can see later for the how they respond. And the second is the individual ecological function. This one actually originally Tongli's paper is right by is another name. Is a individual what's his name? Individual transfer function or something. So Tongli now suggested me just use the individual ecological function. This one is for the site. So include all the provenance. So each variable is the provenance from provenance climate. Then after test the individual response function and also the individual ecological function, we merge these together as we only call it is universal response function. Here the variable could be for the provenance could be the geographic variables and also the climate. For site, usually we use the climate variable, and also like X1I could be several, not just one climate. It can be put 2 or 3 as long as they fit the model. Here I show how much of the variance component for the site and the zone group, also the population. As I mentioned, currently we define the 21 groups. Also here I call the zone, and then the 1100 provenance. Actually, provenance only explained like 2.6%. Left side is the without filtering provenance. Right side is the after filtering several hundred provenance in our regions. For the variance component, they are more or less similar, even with filter or without filter. Site can be explained like 67% of the variations. That means the plasticity is quite large, actually. But zone plus the population can explain 20% of the variation of this bigger population. Here is the individual response function, the curve for each population. I just pick up 3 of them based on the latitude from northern Sweden to the southern, like Bulgaria, even Slovenia. Actually, they can explain lots of, for some of the population, they just one climate variable explained more than like 80% of the variation for each dot, for each provenance. Here, I just want to mention actually it's better to cover like one salt. For this experiment, maybe it's better to plant one more site to the more warm place. That will make this curve even more better. Here is the individual ecological function for 3 of the provenance, before filtering provenance and after filtering provenance. For this function, actually, after filtering, it's make much more improvement in the fitness of the model. So we don't need so many provenance in the upper areas, but maybe we need more for the northern Scandinavia. As we can see from the trial T8, the first one, even with the peak of this curve, even did not reach the the top of the curve. So which means it's better to have more provenance in northern Sweden, like northern Sweden, northern Finland. I use the universal function. Finally, we use 11 variables for the site and also the provenance. The model can explain like 70% of the variation. If we just wanted to show simply show how the model fitted for each provenance dots in each site. Here is use just two climate values. One actually is just geographic variable for the provenance. Provenance latitude can explain much better than the climate because based on the previous model, Zoom group explains well than the provenance. So provenance did not capture too many, just 10% of the variation. And but the GCD5 can explain like 55 or 57% of the variation. Due to here is the sum of the result, we can see this. Here, this figure is the natural distribution of this solution. Then this one is the model we predicted based on model in 1980s. Here, as Northern Scandinavia, we cannot predict based on current data because as we can see, the provenance without the provenance in Northern Scandinavia and also the sites. From here is we predicted based on the RCP 2.5 and RCP 2.85 for the local. Top is the local population, then the bottom two figures is for the optimal populations. The main conclusion from the figure is for RCP 2.5 or RCP 8.5, as long as the temperature, just the temperature increase 1 or 2 degrees for the whole distribution, usually can increase. But for the central Sweden or central Europe, they did not increase, it will be decreased. Within 3 degrees for the northern Europe is better, but for the central it's not, actually. I just make some similar conclusion or result. Site and zoom effect can explain like 60% and 18% of the phenotypic variation, which indicated the spruce has very high plasticity and but slight moderate local adaptation. The model can explain the 72% of the phenotypic variation. The one for actually is GBD5 is okay, it is the best one for site, but it's still as like free frost period also can sometimes explain, also can you explain like 40 or 50 and also annual temperature. But here I just use the GBD5 as the first one. As a modulate climate change, like 1 degree in 2050 or 2007, the height will be increased for this, even for Central Europe, but not for the whole Europe, but not for the Central Europe. But if we use the optimal provenance for each site, then the predictive of the central area and also the whole area actually could be improved 55% and also 30%. When the temperature increased 1 or 2 degrees. But this is just the situation. How much accurate? It depends on the model. So the trend, like increase or decrease, I think more or less similar, even in data improved in the future, like later. So now we are continuing to do because without the fit of the northern Sweden and northern Finland. As we can see, the predicted height. I just wanted to mention we included a new collaborator from the Skogforsk in Sweden and also the Norway, Nibio, to put more trials. Like now, I planning to use more than how many 20 maybe trials in Norway and Sweden. Actually, they were planted similar years. Like for Swedish, southern Swedish trials, they are planted in 1 year later than the Ufro trial. Ufro trial was planted in 1968. These series were planted in the 1969, but less provenance. Left side is the next, those 1100 provenance. Now we covered extra provenance. Here, mostly covered in the Scandinavian area. Then what we are currently doing is we collected the parts and extract the DNA from the one of the northern sites in Sweden, and then one of the south sites in Hungary. Actually, we collected parts from in 2018. This one was northern side was collected. So we have all the genetic data now for these two types. This northern, and we will use a training site, and then we will use the validation site for the central Swedish this Ufro provenance chart as a validation one, and then we will use another family level of the.
Can you try to finish?
Yeah, one, two slides. Because why we selected these two sites is because the northern side has the negative correlation with the rest of this Ufro chart. So we can very clearly to see for the allele frequency, how they change, and how we can use this frequency to predict even for the new family levels, based on the family frequency or family genotype, to predict as long as we know the genotype for each family or each provenance, we can predict how they for the future climate. And this one is the one which we sampled this year for the Ufro trials. We wanted to be considered as validation. I think that's what. And finally, I need to thank all the Ufro provenance participants, and not all. I meant here is just some of them. Also, some of the people they participate in the data analysis and also contribution for the data. Thank you.
Okay, we have time for questions, I think.
Yes.
Yes. I have a question. You modeled the distribution of local and optimal of the Norway spruce, and I wanted to know how did you model these things in 2050 and 2080, if I remember well, for the two different cases like 4.5, 8.5 RCP.
That one. It's because we have the climate DT. So climate DT can support the climate in the future. As long as we build this model based on the currently the climate, and also then we can predict as long as the model built based on the existing measurement age and also this period of the climate, then the model has one variable for the climate. So as long as you put some of the climate in the future, then the model will predict for each position. When we build the model, it's based on the current But then because we have the climate variable in there or geographic variable, then later as long as the climate change, then as you put the climate, it will predict it.
Okay, one more question maybe.
I would have a question for future experiments, because here you take two benefits of an old experiment, Provenance Trial, established by collectivity of researchers in the past. From the experience you have on the results here, what could be your recommendation for new collection of provenance of spruce, and in which environment to re-establish a second generation of provenance tests at a European level to improve the prediction of place and performance of the Norway spruce in Europe?
Actually, when we was analyze those data, we was thinking how to improve, because this one is 1100 provenance, especially lots of the provenance in the outer area. But since there is not very much difference for the altitude, the altitude, so we don't need so many provenance. But if I give me one future, if I am the holder of this experiment, I will just use 100 provenance. Then if we have more money, then just build just one provenance, one block, like in northern cover more northern and more south of the more warm climate data, climate locations. Then the model will improve, but the field work will be less. And then the prediction will be more accurate, and all the participants can be measure the height, the genotype data. Then it's more easy in the future to predict for the whole region of the spruce. It's more simple than now. Now it's too much problem, too less sites. Actually, one block could be one site. Here actually, as we see, the Sweden have the 11 block for each trial, but actually we don't need 11 blocks. 11 blocks could be distributed in the whole Sweden. One block, one trial. One block, one trial. That will be much accurate to cover the whole Sweden area. We only need one trial or one. We don't need so many actually.
Okay, one more quick one.
Yes, it's a very general question concerning this kind of studies based on provenances. And especially one of your conclusions, the first conclusion that says that we find a very high plasticity.
Yes.
And so I'm wondering how we can tell apart the true plasticity from genetic diversity within provenances.
Can you repeat that?
Yes, it's well, you say that based on these curves you're obtaining, so you conclude that you have a lot of plasticity associated with that curve. So how to be sure that this comes from true scientific plasticity and not from genetic variability within that population?
The plasticity and it's not a genetic here as the site. If the experiment decides well, then it's already active. Like a site is for the environment, and the provenance or the zone is for the genetic level. And I'm not sure what do you mean how accurate the plasticity and also the genetics. Plasticity is part of the genetics.
Okay, that sounds like it's a debate that needs to continue afterwards.
Coffee break.
Okay, so I think we move on to the next talk now, which is Katerina Lippern from the Keynote Presentation. We'll be speaking about phenotypic plasticity, full signals of local adaptation.
It's not there. No. Wait, I just didn't, because of the V1 this time. Okay. Yeah. Yeah, okay. If you don't understand me, nicely, please interrupt and tell me. So I'm Katerina Lippern, I'm from the Junior Institute in Germany, and I also want to talk about Norway spruce. And yeah, climate change causes major changes in the composition of our European forest ecosystem and their ecological and economic values. And forest management obviously has to adapt to these changing conditions. But how may forest practice actively set the right decisions in terms of species competitions for the future forests, and may even assisted migration be an option? So we urgently need models in order to make the right decisions. And in the following, I will present you two of such models. I have to click here.
Yes.
So, and that's just where we want to jump in with the first model. So one of such models are adherent-based species distribution models, which predict potential distribution of species based on the correlation between observed presence of species and the corresponding environmental conditions. The most common predictors are climate variables, but these can also be soil or antagonists or whatever, so there is no restriction actually. There are several uncertainties in this model, like the sampling design, the climate scenarios used, the different algorithms. And in order to account for these, it is now common to build ensemble models. And this is here shown as an example for Norway spruce. So we used national forest inventory data assembled by Mauri et al. In order as presence of Norway spruce, which we built then, and then we built the median of 5 different algorithms in order to predict their future range with climate change. We used 4 climatic predictive variables that were species relevant and weakly correlated. So these were namely the high-risk climate moisture deficit, the mean coldest month temperature, the mean driest month precipitation, and the temperature difference, which is a measure like an indicator of continentality. And what you see now here on the right side are our predictions. So the green area is the probability of presence of Norway spruce, and our reference scenario was the 1960 to 1990 normal period. And the black polygon is the natural distribution of spruce.
So you can see that it was distributed quite far outside of its natural distribution, particularly here, where it's planted due to the ease of, to the ease just of doing this, and because of its economic value, it was used largely to reforest degraded areas. But if we look further in with climate change, the species distribution will retreat towards the natural distribution, as you see particularly here, and then it will even retreat further within. So it will remain suitable, especially for that northern latitudes, as well as the higher elevations, like here, for example, in the Alps. So, okay, we can look at this and we can say we're done. We go further, we have our model, and we recommend to not use spruce in all of these areas. But obviously not. Many of us have worked with provenance trials where we have planted provenance sample across large geographic gradients and brought them at different sites in order to plant them in reciprocal design in order to see how they grow there. And what we see is that they're clearly not a homogeneous unit, which is assumed for the occurrence-based models, but they react differently to climate. So the populations are adapted to their local environment where they're sourced from. And what we can see here is tree height of Norway spruce populations at one particular site that vary, but there is somewhat of a trend with a better tree height with a higher temperature where they were sourced. But further, they're not only locally adapted, they also show plastic response to a changing environment. And here on the right, we have three different provenances planted at various trial sites and how they react to a different environment, to the environmental variable at the sites, which is here the summer heat moisture index. So with the low index, they grow poorly. With an intermediate one, they grow very well. And with a very high index, which means it's very warm temperatures and low water availability, they grow poorly again. Trait-based models, which we already have seen in the previous talk, they fill this gap. So they predict quantitative trait expression based on observed trait variation in the field, which may be survival, phenology, or also growth. And they consider local adaptation and phenotypic plasticity, which are the most important processes in response to tree populations to climate change. Prerequisite are large provenance trial series spanning large geographic and climatic gradients. And the statistic models that may be used are, for example, universal transform functions, linear mixed models, or machine learning techniques. Sorry. So we made use of one of the largest transplant experiments, which is actually the same one that we've just seen in the previous talk. And we used the International Provenance Trial of Norway spruce, sown in 1964 and planted in 1968, where they planted 1,100 provenances. And they distributed them to 20 experiments across Europe. And obviously, they couldn't just plant 1,100 provenances in a randomized complete block design at one site. This didn't work. So they used a stratified sampling where they picked these 11 blocks of 100 provenances, so each represented the entire area and can be treated as an individual trial, which are approximately comparable. And they were planted in a randomized complete block design with 20 replicates each, single tree plots, and almost all trials have the same design. And what we've done now, we've sampled tree height also for H11 from the literature which was available, and for 32 site locations, which means 97 blocks of 100 provenances. And you can see them there on the map to the right. I don't can't really get along with this one, sorry. You can see that there's trials from all over, from Sweden, Norway, Scotland, Germany down to Hungary and Poland. And what Jan also pointed out, there's like this difference that some of them, that there's some of them where they're all 11 blocks were planted at one site, like the Swedish ones here, for example, and others, like in Germany, for example, where they're planted at up to 9 sites because of space restrictions. And we also fit a universal response function where we included provenance climate effect, site climate effects, interaction of these two, and also a geographic effect of geography, as there may be other effects as gene flow or post-glacial migration that shape among population variation. And in an iterative model fitting process, we tested 14 biologically relevant climate variables for all site and provenances, as well as latitude, longitude, and elevation and combinations of these. And this year is our final model, so it explains 75.8% of the variance in the data. And it contained two site climatic variables, one for provenance climate, one for provenance geography, and the interaction between provenance and site climate. And let's have a close look at the contribution. We've also seen in this data, obviously, a very high phenotypic plasticity. About 32% of the partial R-square was explained by site climatic variables, which were namely the length of the frosty period and the summer heat moisture index. And we see a quite dominant latitudinal effect, which Jen showed nicely in the previous talk, which explained 14.6% of the variance. And this is most likely attributed to the post-glacial migration to the 3 different domains, like the Baltic domain, which came down from the north in Scandinavia, the Hercyno-Carpathian domain, which came back from the Carpathians to the Beskid Mountains, and the Alpine domain down south. And we had the interaction, and we had continentality as a climate variable for provenance with only 2%, about 2%, and the interaction less than 1%. And these here are our spatial predictions, which came from this model. And the scenarios are the same as before, like the 1990, 60 to 90, 2020, 2050, and 2080. And all area with the positive high growth here is basically like a fundamental niche where species could grow without any competition, without other species. And obviously in some areas very small, so it wouldn't really make sense there. The largest extent of good, but let's better concentrate more on this, on the green part, which is the area of good growth, of superior growth. And it is predicted to be the largest extent here for the baseline scenario, particularly here in Germany, Poland, it goes up here to the Baltic, this area here. So it's quite a large area, and it goes a lot beyond the natural distribution and even further beyond the part where it was planted, what we have seen previously in the Adherent space model. But similarly, with ongoing climate change, we see a decreasing growth potential, which is mainly due to the increase in the summer heat moisture index, so the increasing heat and reducing water availability. And we see that the area of productive growth similarly goes back to the area similarly goes back to the higher elevations, like to here in the south as well as to the north. But if you concentrate here, for example, this part, we see that there is an area where we get a better growth than we had before. And this is because of the elongation of the frost-free period. So if the frost-free period gets longer, the growing season also gets longer, and they make more, they can make use of this. In this way, they grow better. We likewise did predictions planting optimal populations. And optimal here was derived by taking the partial derivatives from the model for the provenance effects. And that way, we identified the optimal population for each kind of pixel on the landscape, basically. And then we predicted for these also the growth that they would experience for the different scenarios. And here on the top, you see the predictions comparable to the ones in the previous slide. But in order to really show what the difference is, here this is the achievable way that can be achieved by provenance choice. And we see that there's a pretty high, there could be a very positive effect in the north, as well as here more in the elevations, but it really may not help, the provenance choice may not help to compensate the retreat of Norway spruce in the flat and intermediate regions, particularly the North European plains. So we've developed now 2 models, and the goal of our study was not only to compare them visually, which you can see here on the right, but we also wanted to actually quantify the differences among them. And in order to do this, One minute. Okay, because it says 14 minutes on the screen. Okay, 1 minute. Okay, so we compared them by chasing the optimal threshold for sensitivity or specificity for the Occurrence STM. And for the other, we developed a productivity-oriented threshold using a regression surface. And we see that there was a reduction of 40% in the 2050s for the Occurrence STM and 55% in the 2080s. And for the trade STM, the reduction was less. So here, I just wanted to show a regional focus where we can see that the 2 models diverge quite much between the 2 models. Like for the occurrences, 98% of Germany will not be suitable anymore for spruce in the future. But with the trade STM, at least in the intermediate, there will be quite some area that is still suitable. And at the end of the 2080s, there's also still some area, about 20% still left to plant spruce. So I come to the conclusions. So both models show substantial retreat towards the species core habitat, so the higher elevation and the northern latitudes. And we've seen a particularly high proportion of variance, which we attribute to the plasticity of the species, because it reacts very, on different environments, it can react quite differently. And the increasing summer heat paired with the decreasing water available was the main factor that restricted growth. Like if we had a summer moisture index over 85, there was just no possibility anymore for spruce. But the frost period, on the other hand, had a positive effect on growth. With the local adaptation to climate was disguised by this very strong latitudinal factor in the data. And the provenance transfer could not prevent the range decline in the center of the distribution, but it may enhance growth within the regions where the spruce will remain, like in the north and in the elevations, the core habitat, basically. Yeah, and that's it.
Thank you, Katrina. Very clear talk. Thank you. Questions? Okay, one.
Yes, a simple question. You compare the distribution map you get with the one produced by Sedel and Al.
Sorry?
Sedel, I don't know, perhaps I don't, Sedel?
Sedel?
Yeah, someone working on the effect of bark beetle.
No,
Spruce,
no.
and he revisited the maps, the distribution maps, including new data coming from the bark beetle attacks.
No, we haven't, there's no data on bark beetles or storms or anything that we compared with this, and it will be definitely interesting to compare and bring these things together, because these are the effects that are causing the large dieback at the moment. This is just like growth at age 11, and these effects that come with, with later age and what we see right now. We couldn't compare them there, but it will be very interesting to get this together. We compared these distribution maps with other already published maps, like visually, and it did go quite in the same direction.
Yes, thank you for your presentation. I was wondering, climate change is not only that we're going to warmer and drier world, but especially to the lower running, more outbreaks of extreme events.
Yeah.
And growth, especially survival, depends more on extreme events, and then particles.
Yeah.
So, then on the average climate. And then again, Spruce has 68 years of rotation, I'm not sure. What's the final age?
It depends on the management. It can be between 50 and 200 years. It depends who is like working with it, I guess.
Should reach 2080, right?
Yeah, maybe not, maybe even not. I would say it makes a lot of sense to reduce the rotation for Norway spruce, to look into very short rotation periods, and then see, okay, how did it behave, and can we go on and even get another generation? But for Germany, what we've seen in these maps, I would say we can make use of one generation of spruce, but just a very short one. We shouldn't wait for 100 years. This is far too long. Then we will get the storms and we will get the bark beetles. But maybe we can let it grow for 50 years, and then we should say, okay, that's it, and we take it now and not longer. Sorry?
There's upper maybe, but not anymore lower.
It's hard to understand with this microphone. I'm sorry.
I said maybe in the upper Sauerland, you
yeah.
You know, anymore. I was wondering, because I think we have all done this exercise of looking for that. Maybe what Catherine asked for, what should be done in Provenance Trials? That's one possible more narrow monitoring, for example, to this trials to see if there are different bark beetle effects. Some are more broad and bark beetle sensitive and so on. Normally, you come to effect every 10 years or 15 years to measure them. If you have money, if you have a project to measure them 20 years later. And actually, the form to set up this trial should be accomplished by more monitored by more continuous and coordinated one, because normally this normal the local partners has money of their not, and then some are measured every 2 years or 5 years, and some of them has been measured for 15 years.
I think really it should be more the quality than the quantity. Like, this is a huge trial. This is very, very huge. And like Chen said also before, we may not need over 1,000 provenances, definitely not. It may suffice 100 or let's say 200 or something like that. It may be completely sufficient. And in order to get like results and to monitor them in shorter intervals, I think it's a lot about not have too many, more concentrate on specific ones, and then also look very carefully in order to get every partner to measure at the same times, and not measure everyone, one at age 10, the other at age 15, and the next one at age 9 or whatever, so that it's more coordinated to have it all at the same time, and then it makes actually very much sense for these models if you have this this same data kind of, because it was quite difficult, or it is for this series, but also for other trial series, to assemble similar age. And we would have really liked to go to a later age, like age 20 or 25 or something, but it's just impossible because we cannot get this amount of data together then.
I have a question. You stress the point regarding potential interest of trait as the model and inference MDL. And regarding trait model, the trait use is high growth. It's performance trait. Have you an idea if such model have been using reproductive traits, the combinative? Because I'm sure that also for the future view of the dynamics of the species, it would be nice to have exactly the same approach using reproduction traits in order to fit and have an overview, because I think dynamics is both reproduction and growth.
I think that's a very interesting idea. This is a very interesting idea, but I haven't seen any of these models that are using fecundity data. Yeah, it has been done for DBH and height mainly for now.
Okay, thank you very much. I think Catherine is lost, so she's got one last late question. But I think we need to move on. So the next speaker will be Octavio Carlo. We'll be speaking about climatic response and adaptation in coral.
Seems that the screen is frozen, so let's wait a moment. Yes. But I can start meanwhile. So I'll talk a little bit about one of our projects that is where you use gradient forest to predict the genetic response of the cork oak. And so you can see there, here, these are the people that, well, now you cannot see, but this is that second slide there is the people that at a certain part of their career work in my group, and these contribute to the results that I showed you. They were essential for the results that I showed you today, if I can, of course. The third slide here are the people that is not from my group, but the people that from other groups and other institutions that collaborate with us, and they are also essential for the results that I showed you. I hope to show you today. Maybe you can use the PDF. Do you have a PDF?
Yes, I tried it.
Okay.
It's too much work about
So I go to the fourth slide, and the fourth slide is the title of the project. And the project, the short name of the project is Keep Pace, and it's a project where we try to see how the cork oak will respond to climatic changes across the Western Mediterranean area. So and that project is financed by the National Science Foundation, the Portuguese Science Foundation, And so today I will show you some results of that, the preliminary results of that project. Okay. So. I hope you discount the time. Okay. Not, I can continue without slides. I came here into the lunchtime, make sure that everything was working fine. So. Let me check in mine. Check. It's okay. So now this is the people that were part of my group, essential for this project. And these are people from other groups and other institutions that collaborate with us. So this is a project, the title of the project, the Portuguese foundation that financed this project. And so here are the aims of this project that are relevant for the talk today. So we are trying to understand the population structure and the demographic history of the cork oak. We have tried to detect the genomic signature of natural selection and which genes and genomic regions are associated with relevant environmental variables, and then how populations will respond genetically to climate change. So here I adopt the framework of a recent review of Kaplan in 4 steps just to make the communication easier. So the first step is data acquisition. And for the data acquisition, so we have this population sample, either from a provenance trial in Portugal, or that we go there and sample the population. Then we obtain GBS genotype by sequence data, so we end up with a large SNPs data set. Step 2, we try to identify the genomic base of local adaptation. And so this is the second step of the framework that I mentioned. And here, what we try to do is set up try to find the association between the environmental variables and the SNPs, the markers. In most cases, we don't find the casual SNP, the SNP that have a phenotypic effect. What we find are SNPs that are in linkage disequilibrium with causal mutation. So we find these associations, and rarely we find this type of associations. We are able to find them because they are in linkage disequilibrium with the causal mutation. So what I show you today is the first some of the preliminary results that are already published in 2 papers, and so this is what I show you today. So the first thing is the structure, and it's essential to then analyze the effect, the detection of natural selection to understand how the population is structured. And this, you see that there is a kind of west-east difference, okay, very, very weak. You can see in the next slide that this is the west, the blue ones, the east are the red ones. And this is the second study the second paper that I showed you before. We find some individuals from some populations in between that show a slightly different pattern. So we identify a slightly stronger structure in the second paper than we detect in the first paper. The reasons for that, I can explain later. Then we did a genotype environmental association analysis, and we find 274 associations, 249 SNPs. Most of the SNPs most of the associations are with these 3 types of environmental variables: precipitation, isothermality, and mean temperature of the diaspora. Then we managed to map 22 of the SNPs in the Quercus lobata genome, so a different oak, a Californian oak. And this we are able to annotate 10 of them. This is the list of the 10. The 2 more relevant ones are these ones that are related, are genes that are SNPs in genes that are related with drought stress or drought response. Step 3, generalizing the genetic environmental relationship. So here, what you use is gradient forest to do this generalization. A gradient forest is a machine learning method, and it starts to establish the turnover function between the SNPs and each SNP and the environmental variable, and that then creates an aggregate function of all SNPs in relation to the environmental variable, and it gives you the importance of each environmental variable in explaining the turnover function. So based on this, you have here the aggregate function for the neutral SNPs and the aggregate function for the adaptive SNPs. So the 265 adaptive SNPs or potentially adaptive SNPs, and then you have the neutral ones. And you see that in some cases, for instance, for this variable, that's quite relevant, you can see here. You see that the response to the neutral pattern across this environmental gradient is quite different for the neutral ones and for the adaptive ones. You can see that for other variables. So when you try to generalize that and put it on a map, you see here 2 results. Here on the left side, you have the allelic composition. And so the color similarity means similar allelic composition. So the green ones here, the browns and so on, the light greens there, and so on, so on, so on. This type of color that I don't really pink one around here in France and so on. So this is the allelic composition, based exactly on that model that I described before. And this is the difference between the neutral SNPs and the adaptive SNPs. So the hot colors means a larger difference between the aggregate function of the neutral SNPs and the aggregate function of adaptive SNPs. What it roughly could mean is that the red areas are areas where there are strong effect of natural selection. So there are strong differences between the neutral pattern and the adaptive SNPs pattern. 4th step, measuring the shifts in the genomic optimum. So for doing that, what we try to do is to measure the difference between the current one. So we establish that relation between the environmental variable and the genetic response. And now we project that for the future and see what come out. You have different methods for this kind of prediction. And the two that I will show you today are the Regression Models, RONA, and the gradient forest. Okay. The RONA is working this way. They establish a linear relationship between the environmental variable, here temperature, and the allelic frequencies, and it projects for the future in this way. If you change the temperature in this way, okay, if we increase the temperature, we expect that the population keep the pace, changing the allelic frequencies, and in this way, okay, so it increased the allelic frequency here. And so this difference in allelic frequency is what the authors call the RONA, the risk of non-adaptance, okay? So this is the amount of allelic frequency that have to change in order that population keep pace with the climatic change. I'd like to emphasize this is a linear relationship. And here you see the results of the data set that I show you. And you see that, for instance, this is for a more conservative climatic change scenario. This is for a more aggressive climatic change scenario. And you see that, especially one population, that is the population of Catalonia, show in both scenarios a dramatic RONA, a dramatic change in allelic, need to change in allelic frequencies. Here is 40%, so RONA 40%. In order to keep pace with the climatic change. And you see that for other populations, the value is much lower, even in the most aggressive climatic change scenario. Here we compare the results, our results, with a more aggressive scenario, with the original paper that introduced the method from Relstab and others. And you can see that the values for, this is for Switzerland, and the values for Switzerland tend to be much higher than the values that we find in our populations. The reasons for that could be either biological or technical. I don't discuss that now, but we can discuss that later if you like, especially in front of the reviewer. Now we go for the other one, for the gradient forest. And the gradient forests have a non-linear relationship between the environmental variable and the genetic response. You can see here that for a small change in climate, in temperature, you have a large change in the allelic frequency, and this is called the genetic offset. And you can have, for the similar type of climatic change, you can have a small change in allelic frequency, exactly because the relation is not linear, is non-linear. So, and here are the main results of today. This is the projection of the genetic offset for the population, for the area that we study. And the red colors mean that the genetic offset is much larger. The yellow and the green ones mean that the genetic offset is much smaller. So here you identify these areas, there and there and there, as the areas with the highest genetic offset, where the changes in allelic frequency will be much larger than other factors. So, sorry. So in this recent review of 2020 of Cap Planck in the annual review of ecology and systematics, they identify 15 papers that work in this issue of genetic prediction. And our paper was one of the 15 papers. Meanwhile, other papers came out and so on. But in the moment that they did this review, only 15 papers address this problem. So it's a very young area with lots of limitations. So since then, there were several papers that addressed the problems of the limitations of the method. And these are probably the three main simultaneously critic and the defenders of the method. Okay, so be aware that there is strong limitations in these methods, and so so what we'd like to do is there are also other methods, as you can saw in the previous talks, other methods to predict climate change response, non-genetic and non-genetic approach. Here you have the ecological niche model or species distribution model as you like. And here you see what happened to the cork oak in the different models along time. And you see that if, for instance, in Portugal, it disappeared from the south Portugal and South Spain, and moved to the north in some years, and it's almost disappeared from the south. Here you see that it's expanding in west of France, so good news for west of France, for Bordeaux and so on, so good news. And if you go, other study that came out at the same time using similar methodologies, you end up even in a more drastic result, is that it completely disappeared from southern Portugal. Okay, it is spanned here again, but it completely disappeared. So our aim here is to fine-tune this ecological niche modeling using this genetic offset approach. So the idea is not to replace this, but try to fine-tune, try to merge the two approaches and see what came out of the two approaches. So the overall aim for us is try to create a scientific basis that can help us in assisted migration or assisted gene flow and contribute to a smart regeneration and an ecological restoration of the cork oak. So questions? If you are interested in this system, this paper came out last Friday. Okay, so it's very, very recent, and I finish and I wait for some questions. And thank you for, in spite of all problems, you stay and you stay to the end of the talk. So thank you very much.
Okay, do we have any questions? Maybe the first question would just be about the discrepancy between the non-adaptation or maladaptation values between your method and Christian Yellstadt's.
Now, the method is the same. The thing is that either they use the difference between the Switzerland results and the Portuguese results is probably due to two main things. One is either a biological answer, is that the intensity of the change in Switzerland, because it's a mountain system, will be more drastic than the intensity of the change here. Okay, so this could be a biological response, and so in order to keep the pace with climatic change in Switzerland, they have to change more. This is a biological response. A technical response is that in the Switzerland project, they use codon regions. So they use coding regions. And we use anonymous SNPs from either regions that are coding regions or are non-coding regions. And so that could in a certain way, emphasize the changes in the Switzerland populations in relation to our population. This could be a technical thing. So I really don't know the two answers, but could be the two hypotheses.
No, thank you for the presentation. I was thinking about the niche modeling and the idea to include the model, the classic species distribution model, the genetic information. Because what we are observing is that if you cluster your distribution according to some rules, for example, if you design bioecological groups, you model each group separately and you sum everything, the result is different. than a classic where some features will be somehow masked by the huge amount. So it was, my question is if you have tried to do this.
No, not yet, not yet. We are actually, this, as I told you, this is preliminary data. We now have better data, more samples, and so we will carry out that, but with this new batch of samples, new batch of results, not with this one. These are quite simple results.
Just a question regarding the use of genetic information and allelic frequencies. Do you know if it could be possible to do more than an approach marker by marker, frequency by frequency? Because probably in population, it's the allelic combination and maybe an haplotypic view, which is the richest. I know that it's quite complex to reach haplotypic information without man, but do you have an idea how to progress on that?
Yeah, the gradient forest and other methods that I show you are, say, community level methods, in the sense that they don't use the information of each SNP, but aggregate the information. So actually you have a multi-SNP result. So you don't have a single SNP result. Yes, you can decompose in single SNPs, but you actually, you have an aggregate function that integrates the information of all the SNPs. Let me show you here. So here you can have the decomposed, so the individual SNP information that you see how each SNP respond to the environmental variable, but you also can have the aggregate function where the set of SNPs, not a single SNP, but the ensemble of SNPs are responding to. This is what you have here, is that the ensemble of SNPs. This is not a single SNP response. This is a batch of 2000 SNPs, the ones that are neutral, and these are the batch of 265 SNPs that are candidate adaptive SNPs. And so this is a response not of a single SNP, but is composed by the response of each SNP, but is an aggregate function of the response of that set of SNPs.
First is about the climate data used and whether there was thought about looking at different climate models, different climate models to investigate the uncertainty. And the second question also relates to the climate data. It would be interesting to see how the models and the distribution of dragons, severe dragons from extreme events. And there is data available on monthly transfers in the future with an ensemble.
Thank you. I am not sure if I entirely understand your question, but our environmental information came from public databases, World Climate, Chelsea, and so on. So this is the type of variables that we are using. And so sometimes they don't have extreme events in the sense that they don't incorporate extreme events, but some variables, like for instance, the driest quarter and the temperature of the driest quarter and so on, in a certain way, incorporate that information. It's not exactly the same, but gives us the idea that in certain areas, the temperature of the driest quarter is much, much higher. And this, of course, will be influenced by extreme events, but this is integrated data for several years. So yes, we'd like to have better climatic information, but we only can use the one that is available, and it is available for our study area. So yes, sometimes there are local information, but you cannot be extrapolated for other areas. I'm not sure if I answer all your questions, but okay, see if we can talk later if you like. I'll be delighted.
Okay, thank you very much. I think we actually have a break now, so I think we'll draw this session to a close. So could we just thank all of the first group of speakers again for a very nice set of talks. Thank you. We'll resume again in 20 minutes, a quarter to next talk and some flash talks. Hi, hi. How are you? Sorry, come down.
I'm going to reload it.
Oh, yeah. Did you, are you just making sure?
I'm just making sure it's short enough.
Okay. Yeah. Oh, yeah. Yeah, good, thanks. Did you arrive this morning?
Yeah, I came to lunch.
Okay. Okay.
So I heard that was better than traveling yesterday. Some people were stuck in France or something.
Flights were cancelled. Well, it's this real extreme weather. There was a thermal gradient yesterday. Like really hot from up to northern Germany. But north of it, there was it's quite cool actually. So Britain was cool. But if you flew across it, I think there were some real problems. That there were a lot of sort of extreme thunderstorms and things. That was quite a lot of disruption.
That was the problem.
Yeah. Yeah, good. Okay. Well, we have a quick break and then back in. Yeah.
I wonder if I can.
Probably just 1%.
Thank you very much for this testimony. I didn't
Excellent. Okay, I'm satisfied.
I don't really do yours.
Sen så glömmer man ju lite bort det där
Yes, I Tongli, just like we are on the breaks with everyone's before.
Hello.
Att det är någonting annat. Fast det är någonting som kanske egentligen kanske inte är relevant när man går söder. Det är sant. Det är faktiskt att känna. Ja, visst är det så. Naturligtvis, om man tänker sig att göra det, och då delar
Can you hear me?
Yes.
Oh, you can hear me. Clear?
Yes,
Very good. Yeah, I have problem to hear the voice from your side. Sound very vague, like in water. But doesn't matter as long as you can hear me clear.
just I have to be front because it's so there was once we go back to the conference. I hope we shall use the right.
Are you talking to me? I cannot catch a single word. Hello?
and then a series of 4 flash talks to follow, and we will finish with our keynote speaker. So starting us off in the second session is Arne Steffen, who's going to be speaking about estimation of phenotypic plasticity in Norway's spruce.
Thank you. Thank you for letting us present our materials here. I'm going to talk about phenotypic plasticity, that kind of plasticity that is induced by the temperature or the climate during reproduction that has been discovered in Norway spruce and that has been worked with for a long time. It's also called epigenetics, but I mainly focus on the plasticity of it. And then we can discuss whether it's epigenetics or not. Norway spruce is our most important northern conifer, and it has a very short and intensive growing season, elongating only during 1 month, from first end of May to the first week of July. It's a very short and active growing season, which is important for the adaptation to the climate. Of course, it's important for avoiding spring frost damages or late summer frost damages and winter damages, but it's also important to utilize the length of the growing season and have a growing season that is match the growth potential and the temperature. It's well known that there's a strong clinal variation in the adaptive traits of Norway spruce. So when you have these common garden tests, and we test the needle injury in freezing tests, for instance, we see these provenance means, they follow clients. And these models, they usually explain a very great deal of the variation. And this is the background for our quite rigid deployment regime with restrictions on transfer. But the provenance is not necessarily a provenance. In the 80s, they discovered that if you produce the seed in seed orchards very far south from clones collected in mid-Norway and move the progenies back from this seed orchard here, they had a changed phenology and adaptation. So they observed that the bud first in spring was delayed, the bud set was delayed by 10 to 15 days, even if it was the same genetic material, more or less. Yeah. And the height growth of these materials increased because they were actually adapted to a warmer environment and utilized the growing season in mid-Norway better. And during the 80s and 90s and 2000s, Øystein Johnsen and his team in Norway did a lot of great work on studying these after effects. And they showed that the accumulated temperatures during the reproduction affected this phenology. So it's the accumulated temperatures during the reproduction, during the flower, during the after fertilization of the gamete and until the seed is ripe, that is giving this changed phenology. They even produced clones with somatic embryogenesis under different temperatures at SAMs. So they had the process going on 18, 23, and 28 degrees, and they could change the bud set of individual clones by 2 weeks. And this bud set is one of the most heritable traits we got. So just by manipulating temperature during somatic embryogenesis, they could alter the phenology of the progenies. This also, yeah. So this is why I think we can for sure connect this to phenotypic plasticity. It's definitely one part of the plasticity. So in 2009, Øystein Johnsen and his colleague wrote this summarized book chapter where they said that temperatures accumulated after fertilization spruce, Gothic, and regenesis of seed maturation changed the adaptive performance of Norway spruce seedlings. And there are interaction effects with photoperiod. They studied temperature the most, but also photoperiod. And this clinal variation you observe in the trials, that is usually very strong and might often steep, might be both due to epigenetics and due to genetics. So the epigenetic regulation is important for fitness and plasticity and should be considered a trait in itself. So in B4EST, we have been wondering how to utilize this knowledge in breeding and in assisted migration and deployment of the reproductive material. Can we predict this changed performance based on the temperature sum of the seed year? And can we assume that this effect lasts for more than 1 to 2 years? Because the studies made in the earlier studies made, they were stopped. Progenies were 3 years and they stopped after they have tested the 2-year-old seedlings. But we have been able to follow these materials for a longer time. So to the Skruppa, which was also a part of this team from the beginning, he studied some of the materials they established. 6 series of experiments established in the early days. And he studied how well this effect lasted after these 1 to 2 years. These experiments were treatments where they had warm, cold temperature in greenhouses, in phytothrons, or by provenance transfer. And he saw that these long-term effects were strong in families that expressed the most change in the early tests. So in this figure here, you can see the growth initiation in spring and growth initiation in late summer after 6 years in field. And you can see that the same full seed families here, they perform differently whether they are from a crossing made in a greenhouse or outside in the outside temperature. And this lasts for 6 years. What you also observe was that there was no change in height growth from these effects, but that is debatable because we have also results showing that height growth is affected by this change in phenolic. So then we wanted to model, kind of predict the change. And what we started using was Provenance Trials established around 2011 and 12 in mid-Norway and southern Norway with provenances harvested in natural forests in different seed years. So it was not the exact families, same families, but it was the same provenance. And it was more or less the same gene pool in different seed years. And one seed year was 2006, that was extremely warm. Here you can see the temperature sum accumulated in 2006. Here you can see the temperature sums in 1970, 1995, 98, which was more normal. 2006 represent maybe the average temperature in 2080. So we made some models, quite simple linear models actually, to kind of predict how much change we can expect. So we made these regression models, and they indicated that for a 100-day increase in temperature in the maturation period of the seed, you have around 1 day delayed growth initiation at age 7 to 8. Model has an R square of 0.3 to 65. 1.1 day later bud set. That was actually a very strong model, R square 0.90. 1 to 2 days later growth initiation at age 7 to 8. And there's also a quite strong model. In most cases, both provenance, the localization of the provenance, the latitude and elevation had a strong effect, but the CDR effect were even stronger, usually. So here I try to illustrate how these different CDRs behave. So the elevation of the original provenance would follow these clines for bud flush, for instance. Here's bud flush, yes. And you have this cline that we know, those from the higher elevations, they need less temperature cells to flush in the spring. Those from the low elevations need more temperatures. But the CDR here equals a variation of approximately 30 to 40 state degrees change in requirements to flush spring. But these provenances were quite rough genetic materials because there is not exactly the same families we studied in different CDRs. So we tried to do the same process in this breeding algorithm in different environments. So we have the same clones here. We induced flowering in 2016, and then in 2017 we did crosses. We transferred all the clones to different sites in Norway, and two of the positions here we had an elevational transect from low elevation to the mountain to see how the natural temperatures affected the performance of the progenies after the same process everywhere. It was a very risky experiment because moving around with these across Norway is not very simple. But we managed to do some process at least. And here is one of the sites, a little seed orchard in the middle of nowhere. And we managed to establish a progeny test from these crosses. Of course, everything didn't go as we hoped. We weren't able to make the perfect factorial mating design because a lot of the crosses didn't work in different sites. So here are the full seed family, mom times dad, and here are the crosses that we tried to do across. So there's a very unbalanced mating design. And it's particularly unbalanced when you get to the cool sites where you have low temperature zones. Around 500 degrees during this season is very low for producing seed. So now we have some results from the progeny tests. And what we see here is that here's one line represents one family, and this particular family has been mated at two sites, 700 degrees and 1150 degrees. And you can see that there's a delay in the temperature zone needed for these progenies to flush in the spring. This is for the mid-Norway material. To make it simple, I made two figures, one for mid-Norway and one for southern Norway, because I wanted to avoid adding photoperiod to these figures. That makes it so complicated. But here is for the southern Norway, and you can see that the trend is the same everywhere. There are few cases where you actually have an opposite trend. So in all cases, mating at lower temperature zones produce a full seed family that requires a lower temperature zone to flush in the spring. Producing a family at higher temperatures, then it requires a lot higher temperature to flush in the spring. Yes. And what was also nice with this new material we produced was that the parameters estimated in the first model based on those provenances I showed could be verified. So we have more or less the same model, even if it was the provenance material or the full seed material. So, in summary, in Norway spruce, the memory of temperatures during reproduction and seed maturation regulate plasticity that gives a long-lasting change to the phenology and adaptation. And the change is predictable. The temperature data, when you use the temperature data from the reproductive environment to predict, you can actually envision how the progenies will be. The adaptive potential of these memories should be taken into account when we do breeding and management. For assisted migration, we need to remember that maybe we don't need that to migrate that much as long as we can harvest climatically updated seed from our seed orchards. We can also consider this when we set our breeding goals, because this plasticity actually reduces our need to do directional selection for phenology to adapt to the climate. We don't need to stress that much strong selection for later bud flush, for instance. And it's also important in terms of genetic variability, genetic diversity, because this plasticity reduce the selection effects. You can see that in mid-Norway we have a variation within the families at the low elevation and at high elevation that overlaps a lot. Even if the environment is very different, the genetic variation in the stands are big, large. And this is kind of kept, it's preserved by this plasticity. And here is an example from what we can see in the nursery. Here is, I don't know if you can see that, but this is actually just one provenance. It's from, everything here is from one seed orchard. But these darker plants here, they are from a cold seed year. It's from the Untitled Seeds orchard in 2015, and you had 1100 day degrees this year. And here are the seedlings from the warm seed year in 2006. So this is just a commercial production. It's not a very good experiment, but it's an example of how we can actually see this. So the light green color here indicates that these are still in the elongation. And these from the cold seed year, they have stopped growing and starting to harden earlier. So, of course, these effects are very important in Norway, where you have a short distance from high elevation to low elevation. You can have Norway spruce forests at 1000 meter and at 100 meter with just a few kilometers distance. So when you get this mix of pollen from high elevation and low elevation, it's of course important that the spruce have this kind of ability to remember the climate where the seed was produced, because this will not fine-tune, but help in making the population more adaptable. Well, that was that. Thank you.
Any question?
You have knowledge about epigenetic sign regarding response to extremes?
We don't have any other data than those we have produced from warm and cold seed years. It's only warm and cold seed years that we have studied. But of course, this should exist in other species, and I feel that when we have worked with it so long, I think it's time to start studying drought, other extreme events than just temperature. Maybe we could induce drought in our experiments to see how that affects the progenies.
The first I'm heard about this phenomenon was in 2000, I think it was 2008, the tree reactor in Madrid. I'm sure it was also in 2010. And actually, in the same meeting, I was reflecting on my own species so far. It's a very homogeneous, genetically homogeneous species, and actually we found a very slight differentiation between more inner or more northern provenance that are growing earlier at the time. So that are growing a little bit earlier than with more water, so they're growing some centimeters more, and so it was the same phenomenon. And then I was wondering, okay, if there's nearly no different genetic variability, maybe it's only other effect. We never tried because we didn't get this. But actually, this information were general species. The question all about local adaptation and maybe because it might be masked or this adaptation to the embryogenic memory and so on. So actually, it's special because a lot of things about local adaptation. So maybe even local programming by stress them by greenhouse and prepare it for further warming. So I think it's just interesting topic to research in other species, because actually what we take as genetic adaptation might be just programming process or epigenetic change.
Yes, I'm quite sure that there are. We have to think of both the adaptation and the selection. But I think for the short distances that we see in Norway, where we have this strong adaptation with innovation, for instance, which within short distance, I think this epigenetic memory is more important than the local adaptation. But on the long distances, then I think maybe adaptation and selection is more is the stronger factor, maybe. But then, of course, there's this is probably a trait under selection itself.
Fantastic presentation, very interesting topic, at least for us. You made a very good review of the systematic search for causes for this effect, very convincing, particularly the study with the embryos, which I think it would at least make a strong case of a genetic kind of. How much is it known on the pre-zygotic and post-zygotic selection in fertilization? Isn't most an easier explanation here that seed collected from a whole year would, for instance, have some sort of pre-zygotic selection on pollen growth? So the genetic characteristics of the different crops could be demonstrated as this line of the investigation ruled out.
Well, I haven't participated in the research on ruling out those causes, but Mr. Nielsen and his colleague did. They worked systematically on ruling out all the possible selection stages earlier. And they were convinced after these studies that it wasn't high enough selection intensity at those stages to explain all this change. Yeah.
the temperature relationship and seed variation. There's evidence that the temperature fluctuations, the low temperature fluctuations, occur on cycles, and a decadal cycle is observed. Colder years and warmer years and colder years, because there's a slower increase in temperature underneath all this as well, and the stochasticity of the annual variation. So how should we think about taking this approach? Quite complex.
Yes, it's quite complex, and I don't think we will be able, like, I don't believe like in breeding for a perfect plant, because we have all this plasticity and we have the genetic variation that is on our side when we do breeding. I would ask the question back, could we imagine that this Norway spruce flowering phenology, that is kind of, it's initiated one year and then you have the abundant flowering the next year. Could that be an adaptation to these cycles you mentioned? That when you're in the warm period, it's likely that next year it's also warm and good for seed production.
Maybe when the trees are still young and still trying to close down progeny which contribute to the establishment process. In 10 years or something like that, most of the frosts.
Okay, thank you. Maybe we are going to go to the flash presentation. First, Martha. Martha. Thank you.
Yes. Okay, hello everyone. I am gonna present to you the response to selection in an aerial coniferous bank. So to start, I start with the general question about how plants modify the phenotypes according to the environment. So generally, when using open pollinated progeny trials, people used to select seeds from a single or multiple populations to install an experiment under the same or different conditions. And as we all know, different responses we can find in each species will depend on a variety of factors. So in our case, the originality of our approach is that we collected seeds from a single population, from the same population, but from different cohorts of age. So we could call it like a multi-year progeny testing. So our aim was to determine whether environmental variation among years, seed mass and maternal conditions had an effect as potential sources of variation on early seedling performance, reflecting different responses to selection and heritability estimates. All of this in the context of serotiny, which consists in the ability to retain seeds in closed cones for a long time to form an aerial seed bank that can be released after an environmental trigger that normally is fired. So this way, we were able to do a retrospective analysis. So we sampled 20 mother trees from a natural population of Pinus pinaster located in the northwestern Spain, as you can see in the map. So for each tree,
for each mother tree and each cohort, we measure all of this information, the tree age, basal diameter, tree ring width, and also we included in the analysis a drought index that is called SPEC. So in total, we had 12 cohorts of age. So we collected seeds, and after that, in a common garden experiment, we measured plant height and plant height at the ontogenetic stage 4, so ontogenetic development. We ran different statistical analyses just to estimate the variance, just to explore in detail the genotype by environment interaction with linear mixed models. But here today, I'm going to present you just the results about the reachability and the response to selection. Just to say firstly that we didn't find an effect of the cohort by itself on seedling performance, but however, we found an interaction between mother tree and cohort, as we can see in this graph. Each line is a mother tree, so we can see that the seedling performance is really different between cohorts. So this could suggest that some effects at the micro scale could be operated. So after doing the other analysis with respect to the reachability, when fitting the general model, we found different values between cohorts, as you can see. After that, when fitting seed mass on the model, we see that the values of reachability decrease. So in comparison with the general models, the values decrease. So finally, we fitted another model with the other maternal covariates, and again, we see that the values decrease, but the key point here is when we included seed mass. It's really relevant. And with respect to the response to selection, again, we see different values between cohorts of rates. We didn't find an empirical response to selection, as we can see, because the values are really small. But the key point here is that we found different responses between cohorts. That this could suggest that we can find variability to face possible future snakes. So just to finish, we can say that we found a strong and widespread interaction between mother tree and cohorts for seedling performance, which implies that the effects of the maternal genotypes were different depending on the year when seeds were formed, that when discounting maternal effects, reachability values were more stable and decreased. That the most informative part of these results is the variation between cohorts, and also that the transgenerational effects by promoting phenotypic differentiation could have a role in stabilizing selection. Thank you very much.
Yeah, this one? Yes.
Hello, I'm Laura Guillermin. I am a PhD student at the University of Oxford, and I'm going to present a bit of my project, which will be basically about the genetics behind the continuous cover forestry approach. And in particular, I'm going to try to answer if these UK plantations hold enough genetic diversity to face environmental changes. As a bit of a background, continuous forestry management has different principles, and one of them is that it will relay natural regeneration. And the thing is, these UK plantations are going to be transformed into this more sustainable and diverse type of stand to try to cope better with climate change and other challenges. So I'm going to focus in the fact that the primary, the canopy trees could, which is our hypothesis actually, is if these primary canopy trees hold enough genetic diversity to face the current and future disturbances. So we aim to assess the diversity in the gene pool, and we want to study how it's transmitted to the offspring. So the big picture of my project, I have 5 sites in England with several sites in each of them, and I'm looking at 2 species that compose these stands. Is Pseudosugamensiesi or Douglas fir, and the other is Tuya plicata western red cedar. And the way, or the technique I selected to use to genotype them is Fluidium, SNP type assay, or allele specific PCR, which I'm not going to go into detail, but basically what I did was to filter a big data set of available SNPs for Douglas fir. Then I tried 158 SNPs in a small sample of the bunch samples. And then I actually genotyped 72, with 72 of them, which are the results that I'm going to show you. So it's a very preliminary results. As a quality control of these first 72 SNPs that I managed to genotype, well, first of all, this is just for Pseudosugamensiesi, and just one of the sites, so it's 40 adults and 140 juveniles. So I wanted to check how they cluster. So this is an example of a very nice SNP, how they cluster. Then not all of them clustered this well. Missing data per individual. Only 5 samples show more than 20% of the missing data, and just 2 locus show more than 20% of missing data. So that's quite okay. The distribution of the allele frequencies is also very nice. And this is just a first preliminary population genetic result. As I said, I just put this together very recently because I just made to work the genotyping protocol. So if you have any comment or anything, I would really appreciate it. I defined 2 groups, adults and juveniles, and in the PCA that I made, there's no difference, although the shape of the cluster maybe could be different in a way. And then by checking the genetic diversity, there's no difference between the 2 groups, although the observed is lower in both than the expected, which could be due to inbreeding, but it could be also the other things. So I will look at that in more detail in the future. The next steps, of course, is to finish genotyping all the stands I have for Douglas fir. I will also want to look at different set of SNPs scenarios by quality and how they cluster to see if that changed anything in the results. And of course, measure genetic diversity. And then for Tuya plicata, I didn't talk about it now, but I already performed genotyping by sequencing in some of the samples. I did the variant calling, and the next steps will be to genotype and to measure as well the genetic diversity. And that's all for me. Thanks to my lab mates, my supervisor, John Mackay and Gary Kerr. And I also wanted to take a second to remember Pablo González, who was my supervisor during my undergrad, and he passed away in April, and he was a great researcher in forest genetics as well. So I just felt like saying it. And that's it. Thank you so much.
Okay, ladies and gentlemen, I have been given a task to entertain you for the next 5 minutes. Good afternoon. And as you have noticed from today's presentations, there are one group of people that is extremely excited about dead or dying or damaged trees in the forest, which is forest scientists, of course. That's why we have a pile of presentations about the ash dieback, for example, and that's why we have a pile of presentations about Norway spruce that happens to be sensitive to whatever is thrown at the Norway spruce. And therefore, for a contrast, I have chosen a small data point about Scots pine, which seems to be not sensitive to anything and is slowly growing and producing nice raw material for our bioeconomy across the European continent at the same scale as Norway spruce. So just to see where our data points are, the project is notably smaller in scale, I guess by a factor of 2 at least, notably smaller in scale as B4EST. But still we managed to cover some set of provenances located in different points. And as you have seen during the presentations about the gross climate response studies, today the one approach would be use a certain age, a certain trait, and then assess the effect of climate on that trait and the provenance variation. So what we have tried to do is a slightly different approach. We have tried to combine the dendrochronology, which basically is studying tree rings and the response, depending on ring width or the basal area increment, on the response on the different meteorological factors and provenances. Provenance research at the same thing. We have selected the number of sites and the number of provenances located in those sites, thanks to the previous work of the forest tree breeders before us. And you can see the basic data about the number of the material and the variation in the slide here. And we have used the individual tree data as a data points for further modeling, basically detrended the dendrochronology series, assuming that each trial represents a relatively small variation in the meteorological conditions. So we can say that this is the linear relationship within the trial or within those years that the trial is representing, and then using the values and using bootstrap regressions to get the middle points for the slope coefficients, and then using this all in the calculations. As you can expect, and as you know from your studies also, that there are provenance differences in the response that are far from linear in the response to different variables, gross response to different variables. That is shown also here by each provenance represented by different color of line. And what maybe is more interesting is if you are trying to look to the heritabilities of those differences, and there are those significant meteorological variables and their heritabilities calculated based on the linear model, as you can see there, with the test site and block within the test site being a fixed effect, and then provenance being the random effect. And also the coefficients of variation. So what does that all indicates? I would say not really proves at the moment, but indicates there are a notable variation in sensitivity between the material and the sensitivity what we can actually derive from a dendrochronological analysis, adding to the information we can know from the large scale trials. So we can better understand our genotypes and do a better selection based on this additional information. So that's it. Thank you.
I thought I was going to be giving the very last talk today, so I thought I would strip out anything complicated, or I thought I'd give you a nice easy one to go through. It turns out there's another one after this, but anyway, this is a little bit of data from a paper that we published earlier this year. So I'd encourage you to either come and talk to me if you want any more information, or to look it up as well. But it's basically concerning phenology. So I'm interested in, well, we've been looking at association genetics in Scots pine, and specifically we were interested in, rather than just looking at growth traits, also considering other traits. And actually, Arne's talk's given me a lot to think about because, yeah, anyway, phenology. So the timing of recurring seasonal biological events, and they have a really big impact on timing and duration of growing season and reproductive traits. And if you plant trees outside their native range, you can get a mismatch between the phenology and their environment. And although a lot of work has been done on phenological prediction, it's based on environmental variation. However, it's known that there's a really strong genetic component, significant genetic component as well, as we see from common garden studies. So what we're interested in is to see whether we can identify SNPs which are associated with phenology and use these to build predictive models for this trait. One of the main reasons that we're wanting to consider this is because survival and fitness can be affected. And so being able to predict for a trait like phenology has a lot of implications for, for being able to predict whether they will be able to grow very well in different climatic conditions as well. So yeah, you can see that, as Arne also mentioned, that you have this really strong gradient between latitude and frost damage. And this is among species, but you also see it within species as well. And so this is just phenology spring bud flush in Scots pine. So you can see from the left to the right, you can see as it progresses through the season. So this is a period of usually about 8 weeks or so, and we categorize it into these different stages. Therefore, it's categorical, it's qualitative rather than quantitative, which have its own challenges as well. So as part of this study that we did, this is a kind of overview of what we did. So first we actually used this middle one. We used 3 species. So we were testing to see whether you could use multi-species when you're doing association trials. We used Pinus mundo, Pinus sylvestris, and Pinus leucinata. And we got the phenotypes, so in this case, growth and phenology as well, and the genotypes, and these were all grown in a glass house. And we did association analyses, and then we took these sets of associated SNPs, and we used these to build predictive models, which we assessed internally using a proportion of the trees as a training set, and then we also tested them using a different set of trees. And these were then independently assessed in just Scots pine as well, Pinus sylvestris, using a different set of trees. So I'm just going to report a little bit of this. It's quite big, and I've tried to strip back a lot of it to fit in. So the very basic summary then of what we found that there were 42 SNPs which were significantly associated with growth. And almost double that which were associated with phenology. But you can see from this that the majority of SNPs that were associated with growth were common, whereas the majority which were associated with phenology were rare. And this here shows the percentage which are assigned to different functional groups. So this is growth and development, reproduction, and response to environment. And then this is one where we couldn't assign where the SNP belonged to. And you can see that generally speaking, they're pretty similar. We see that the majority are associated in proteins which are related to growth and development or response to environment, and not as many associated with reproduction, which is what we would assume to see, expect to see. You can see that the phenology traits here, the SNPs associated with phenology, there's more that are associated with response to environment, which again, is what we might expect to see. And we built predictive models, and these are the results for the internal assessment. And you can see that the results are generally pretty good. We get significant associations, a huge amount of error, obviously, but significant associations between the observed and predicted for both growth and blood burst. So it shows the potential of this approach. It has challenges as well, but I don't have time to go into that now. So I'd just like to say thank you to our funders, and then this is the paper here if anyone cares to read it.
Okay, thanks. Questions?
I have a couple of questions. The first one is for Marta. And I want to know that if you have seen, I remember that is a continuous cones, so there are some cones that have been in the tree for several years, while others are like very young. I would wonder if you see any effect of the age of the cone in your performance in the experiment. She said no.
You mean if I found an effect of this age?
Yeah.
Right? In the
trace.
Because I test, for example, for pasta in porcelain cake. And I didn't check for that. And in the models that I got.
And I have, if I may, and my second question is for Laura. So I did notice, like, I don't know, maybe I missed something because these glass tops are crushed, so it was very fast. But when you put the graph of the allele frequency and your feeds, so, and I saw that most of the things have an allele frequency of 0.5. So that means that the chip where you
No. It's okay. Sorry.
Okay. In that case, I wonder that because when you are comparing, then you are comparing to diversity or common. So it's very difficult to have differences like that. It would be more expensive that if you have any difference. It would be more because there are some rare sleeps or frequency sleeps in one population are fixed and the other are polymorphic. So I don't know, maybe if you, I see there is also some low frequency sleeps, so maybe if you explore a little bit how the frequency you can estimate the comparison that we may find some differences.
Thank you for the question. I think that's also something we want to look at because from these 28,000 data sets we wanted to get the number of schemes to generate. So we put basically this number quite high, so that's why we pay a really high in that way. And we decided to do that we can find these needs ourselves. So we wanted to be sure that these needs were amplified in our population. So that's the first approach we followed. It would be interesting to maybe lower a bit that feature.
Yes.
Thank you.
Thank you.
I have a question for Marta again and for Alice, because I saw in your slide you have used the same index to characterize science. And I like to ask you both, according to which period it was stabilized and why.
I use spade for spade 5 months from green spring because we wanted to know if it has an effect in the environment when it's in the hole. So this space is really important. So in fact, could be operated via and I think okay.
Monthly figures because that's what we were going for all the variables like the presentation. But then we of course have some variable. We have tested a number of drought indexes. This is just the one that we showed from finally. But for the technology, we were going through all the variables on the monthly basis across the years, and then
the state standardization, the use of several slides, but we still standardize according to the side of the move within each site.
Yeah, it could be, but yeah, I couldn't comment on that, I guess. But we have tested several character variables and indexes character and trying to get into that. Yeah.
Questions? We have 5 minutes.
One question to Altair. If I understand it correctly, the sort of the cohorts and the cohorts times smaller interaction that is based on the effects which could be due to, for instance, mental environment or effects or something like that. But in your case, when you sort of have free pollination, have you considered the possibility of sort of differing mating dynamics across years? Is maritime find a very stable flower, so that you can assume that the mating dynamics across years is basically similar, or would there be any possibility that there is something about a father effect timing that he's close?
I have 12,
Did you have markers?
But in that case, we really didn't find different differences in the environment. So between codes, contrasted codes. So in this case, we see even when the values for the estimated values for what were very similar, there was a strong interaction. So then we run the analysis introducing conditions and so that's I don't know if you have.
Oh, thank you.
Yes.
Now I have a question for Anika. She doesn't escape without a question. So I just wanted to know, like, I understand you have the association for the three species, right? So, any common association?
Yeah, that's a really good point.
So we did kind of different species groupings. So we did one with Pinus and Pinus snapped together as part of the Pinus complex. And then I did one association analysis, just so that's just by itself, and then one with all three species together. And there weren't any SNPs in common between the Ruber complex group and the Sylvestris.
More question?
Okay, no, that's the last
I think we did. I have a feeling that heritability was, so there was significant variation among families, there was heritability there, but I don't think it was really high from memory. It was good. But I can check. How young the trees? So the one that last specified was 6 years old, I think. Yeah, so okay, so the point at which they were measured they were maybe much more practice than probably the trees, yeah.
Yeah, because once we measure the last actually for us it's like 15 meters high, the carbon branch. Finally, we didn't get it. Because we use adult, like 15 years old, so we haven't get
Yeah, I mean, we're seeing, so we have trial Scots Pine Road, which is about 50 years old, and we still see really strong differences, a different trial, but and yeah, they've been growing outside, well, 10 years now. And we still see really strong variation in blood bags year on year.
For the GWAS, how many?
For the GWAS, for the efficient analysis.
Yes.
So Scots Pine there was about 150 trees. For Pinus and Ruber there was about 120, and I think for Pinus and Sylvestris it's about 200.
Is those specifically selected or just the
So it was a SNP array. So there was about 21,000 SNPs, and that was reduced down to those polymorphic from memory was 15,000.
So it's so many SNPs. I saw the lot of SNPs. Actually, it's more than like 1 week.
The work found to be significantly associated.
But maybe so thanks for the speakers for bringing time for all the questions. And then now it's the keynote. Tongli Wang.
Okay, so it's time for our keynote. So thanks very much to our speaker, Tongli Wang, who is speaking, is very patiently held on for us from Canada. And we've got our, yes, we've got our timing right, I think, which I hope is good for you. So thanks very much for waiting for us. We were just wrapping up a few interesting questions from the session. And now, please, can I hand over to you for our keynote presentation.
Thank you very much. Can you hear me clear?
Yes, very well.
Great. Okay. Thank you very much. Good afternoon. And thanks for the organizer to give me the opportunity to share my thought and some studies of our group in climate change mitigation and adaptation. Here is my talk, using forest genetic resources for climate change mitigation and adaptation. Okay, here's the outline. I'm going to talk about climate change impact and the potential genetic solutions, and the climate niche models, transfer and the response functions, and the landscape genomic approach, and a summary at the end. Let's start with the first one. So the climate has a big impact on forest trees, no matter change or not. So first, climate is a major environmental factor affecting the performance of our trees. So it has an environmental effect. And it is also a major evolutionary forces driving speciation and local adaptation, which results in among population variation in trees. So it has genetic effect. That's why different forest types are growing under different climate conditions. In other words, the nature has created different forest types for different climate conditions. Similarly, at the species level, different species are occupying different climate conditions. So we can also say that nature has created many tree species for different climate conditions. At the population level, climate also a driver driving the among population variations, create genetic clines for the widespread species, like large pine, as I showed in the figure. So the species can grow, reproduce, and grow wild within their climate niche, within the range of climate conditions. We call that climatic niche. And within that, there are variation among population variations. So through long term of evolution adaptation, species are adapted to their local range. And the climate change is causing a mismatch between the climate that has been adapted to and the climate those trees will need to experience in the future. So for the trees left outside of their suitable climate conditions, they will compromise their productivity and become vulnerable to insect diseases and the natural disasters. On the other hand, there will be new area become available for the species, but due to the limited rate of migration, and it will take the trees may not likely able to catch up with this change. Here, the aspen, large-scale aspen dieback, probably due to the is a good example of those climate mismatch. So the research questions about the climate mismatch and the possible solutions. Many people are interested to understand the evolutionary mechanism of the trees in adapting to the changing climate, or specifically the mismatched climate. This is an important aspect to understand the mechanism. However, it is a completely difficult question. It will take quite a while to get there. And thus, it's not likely we can take control of the evolutionary process in near future. Many are also interested to breed more adaptive trees. This can be through conventional tree breeding programs, and it can be done. People are exploring the approach in genetic modifications. and also epigenetic approach as a merging new way to improve tree for certain trees in a more quicker way. Just like a study shown a moment ago, showing the Norway spruce, this kind of maternal memory is kind of epigenetic phenomenon. And that many people also interested to study how to help the trees to match their suitable climate conditions. So nature has created many species and the genetic clans for different climate conditions. This to help the tree to match their climate condition probably be the most effective approach to mitigate and adapt to climate change. However, assisted migration is still facing resistance from the strong idea of using local and the native species. Many people don't accept that. However, we need to be aware that the local species or local population, they adapted to their local climate, not local people, not local topography. So if the local climate move away, then the species still there is not in their local climate condition anymore. And thus, the matching their local climate should be considered as using local species, although we need to move to different locations. So in a way, yeah, and using to match the climate for species is we can consider as using climatically local species or populations. Climate is changing when our conception need also to change. So the mismatch can occur at two levels. At a species level, here horizontally shown, is the climate niche, the mismatch. And vertically, it shows among population variations. So even within the climate niche and among populations variation, and the population within their climate niche may not in their optimum climate conditions anymore. So, the climate matching need to happen also at the two levels. At the species level, we use climate niche modeling. At the population levels, we use transfer and response functions. Let's start with the climate niche models. So for species, given species, if we can build a climate niche model, then we can predict the spatial distribution of the species. Then we can also plug in the future climate to project their suitable climate spatial distribution of the suitable climate conditions for future climate. After lay down these profiles, it will help us to understand how the natural population responds at different section of this profile. So there is a leading edge and the overlapping edge, which is good for adaptation, and the lagging edge, which the populations are in danger. So this can provide the basis for us to formulate conservation and assisted migration strategies, adaptational strategies. So for the leading edge assisted migration, probably the major approach. For the overlapping area, the in-situ conservation assisted migration at the population level is the right approach. For the lagging edge, ex situ conservation and introduction of new species should be considered. Some facts about climate niche models. So a strong relationship between the realized niche and the climate conditions is the basis of the climate niche models. The assumption is that the realized niche is a result of long-term adaptation and the interactions with other species during the past. And the major challenge for the climate niche model is the model accuracy. So the model, this kind of model is relatively easy to build. The occurrence, which based on the occurrence data, which is easy to obtain. And also there are available algorithms to use. And so to build an accurate model is the key. And also, even you have an accurate model, the future climate is uncertain. So the future projections naturally is uncertain. And another issue is what we can predict is realized niche model. Realized niche is that fundamental niche. The limitation of the realized niche is that even you find the trees are left out of the realized niche, but they may still, within the fundamental niche, they can still survive, grow well. So it'll be good. We can also predict fundamental niche. So this is a limitation. Although those approach has been widely used, but it faces a lot of criticism. For example, the rate of migration, interaction with other species, impact of pests, diseases, interaction with microbes, and among population variations. However, those criticisms are based on how we interpret the model predictions. If we say our model prediction is species distribution model, which has been used by many people. Most people are saying this is a species distribution model. So in that case, all those criticisms are valid. In fact, the model we built in this approach is ecological model. If we say that this is a ecological niche model or climate niche model or bioclimate envelope model, and which predict the suitable climate conditions instead of the species distribution per se. So in that case, most of those criticisms are not valid. Only one is the amount population variation indeed is not reflected by this kind of models. And another issue exists for this kind of model is the extrapolability. So people say, although you can build an accurate model, but can that be extrapolate to future climate conditions? That's a question. And this study shown here is apply a smart way to validate this. They use a large pine, Douglas fir. They use the Douglas fir occurrence data in North America, the native range, to build a model. Then to validate model, use the occurrence data from Europe, because this species has been successfully introduced to many countries in Europe. And their results are disappointing. So they found only a small fraction of the areas where Douglas fir successfully introduced are predicted to be suitable. So their conclusion is the SDM can be misleading. When I see this paper, I also done this modeling. And my results showing that the suitable range can be successfully predicted when I compare with the occurrence data from the Europe for this species, and the matching rate is nearly 90%. So I have a different conclusion. So this model is highly accurate. So what was the problem behind it? So I followed the original paper, and I used scale-free data. I will come to that later. But the paper, that study used 4x4 graded data from world climb. So I did the same, switch it from scale-free data to graded data. My results, the accuracy, model accuracy reduced dramatically, but still higher, but still, yeah. So the difference between these two kinds of climate data is substantial. So the grid data and the scale-free free data, which reflect climate conditions for specific locations. Here, the grade, only one value for each tile. In the mountain area, 4 kilometers grade, the climate can differ several degrees for mean annual temperature. And we found a similar situation while we're modeling the ecosystem classifications in BC and Canada. And if we use the graded climate data, this is the ecosystem. The left is the ecosystem mapped. Here is our predicted on the right. So you can see the accuracy is pretty poor. But if we use the scale-free climate data, and the prediction accuracy can dramatically improve. So then this question, where to get the scale-free climate data? So for North America, and Climate NA we developed. We also developed a version for Europe, and last year we published a paper on this. Several colleagues have used this model. They found that the effect is very good. And then now this B4EST project developed a new version, our version using the same approach. So now come to the transfer function, transfer and the response function. So we mentioned that even within the climate niche, the population variation, among population variation is substantial. We also need to care the mismatch at the population level. But using transfer and the response function. However, those functions require provenance trials. Here I use the lateral pine provenance trial as an example. And this trial was built with 140 provenances, range-wide, and tested at 60 test sites. Very comprehensive provenance trial. And all the populations, many populations are tested at each test sites. So for each test site, the climate conditions are considered the same. For that, we can build a transfer function based on the transfer distance of the seed source in terms of climate. This here is a mean and the temperature, and they use height as a dependent variable. We build this transfer function to reflect the transfer distance on the performance. And we can also build gene ecology function. Instead of using the transfer distance, we directly use the source climate as the independent variables. And the results are about the same. but the gene ecology function is seed source explicit. So a transfer function or gene ecology function represent a local adaptation because they are tested under same climate conditions. All the differences are due to their genetic differences because they are from different climate conditions. Those functions work well for the size tested, and because the same set of population, if you plant at a different test size, the function can be different. So we build all the gene ecology functions for the 60 test sites. Here I show 6 of them. And because each function only represents for the test size, and to generalize this transfer function, a general transfer function was proposed. And in that, you pull all the data across size into one and calculate the transfer distance and the related to performance. And this model performance is generally poor because the feature at the individual size are all canceled out each other, not all, but largely cancel out each other when you pull them together. So many general transfer function has been built for different species, and in general, they can explain about 30% of the total variation, which is not very good. On the other hand, because each populations are tested at different environment, different test sites, we can build a response function. So we use the site climate as independent variables and the tree height as a dependent variables to build this relationship. The response function represents environmental effect on the performance of the population. Same genotype, different environment. So those relationships represent the environmental effect. It also reflects plasticity of the population over a range of climate conditions. So same genotype, different performance, this is a plasticity. The response function is more straightforward for climate change adaptation because the climate change is changing environment, and the response function is reflecting the environmental effect. So it's a perfect match. However, the response function requires many test sites under a wide range of climate conditions, which is often difficult. So in many cases, for many species, they have a Provenance trial only with a few test sites. This is very common. So in that case, it's not able to build a response function. And if we compare the response function and for each population, put them together, we can see that the populations for larger part, at least from the north, and it will generally grow slow no matter where you plant them. So they are adapted to a slow growth for harsh environment. The one in the south do not perform very well either, because in the south, other species push them to the poor conditions. And from the central distribution area, it has the best performance. So in that regard, at the beginning, people think, okay, the productivity in the south will reduce in the future climate, but in the north, it will increase. So it will compensate the loss in the south. But from these results, we can see that the north, you cannot count on them to grow faster than the one from the south. So we have response functions and gene college functions. Response function, each function represents one population. The gene college function, each function represents one test size, but we have many populations, many test size, planting size, and so those function has limitations. So we put them together and develop a universal response function. And here's a simplified version with one clan variables. So this allows us to predict performance of any population or any provenances at any test size. It's a continuous one. It also has an advantage to determine the genetic effect versus environmental effect because they are in the same function. We can use take the first order derivatives to reflect the rate of the change. So we can use this function to predict if we use local seed for different climate conditions. And we can also use the optimum populations for the different climate conditions. So you can see there's a great potential if we use if we do the climate match at the population levels. This universal response function has been used in many cases, and we together did a study with Douglas Fir in Austria, and we built this function and predict the performance and to determine where is the suitable seed source. And then we apply this one to predict entire Europe. In many cases, the prediction is pretty well, although the function is only developed based on the results from Austria and the part of Germany. And additionally, we also use this one to predict the distribution of the native in the native range in North America. Here is the distribution occurrence of Douglas Fir. Here, if we use the occurrence data from Europe to predict the presence in North America, we can only find these are only from the coast. The many parts are missing. But if we use the URF, whether to use height or basal area, so the prediction is much closer to the real observations. And very recently, Lippi and others used the universal response function to define the plasticity and the local adaptation. And later on, after the URF is developed, later on, latest proposed universal transfer function using linear mix effect model. So the objective is to overcome the problem of small number of test size. You know, the URF require a reasonable number of test size, but for their case, they have only a small number of test size. They also trying to introduce the random effect to improve the model accuracy. So the predictor include transfer distance, that's why they call transfer function, and the provenance climate. And this together actually can determine the test site climate. Then the interactions between the transfer distance and the provenance climate. Then they include some random effect of provenance, site, and the block. So we tested this UTF with lateral pan data. And we found that the URF and the UTF have exactly the same performance if random effect is not included. Now, do we need the random effect in it? Although this is different, the URF, you can see that has a provenance climate, site climate. The site is, both are explicit. We can see what is the site climate, what's the provenance climate to determine the response. But in the universal transfer function, you don't have the site climate, you have a transfer distance. So the site is not clear. It's a kind of difficult, a layer of difficulty. So the random, another thing is the random term increase the model accuracy, but not the prediction accuracy. Because when the model has a random effect, when you predict for the new conditions, the random effect has to set to 0. So the random effect is not used in predicting in projection for the future climate conditions or for spatial predictions. The only can be used for the specific test sites. Another thing is the random terms are expected to improve the parameter of the fixed effect. And the purpose is to overcome the issues of small population, a small number of test sites, and improve the prediction accuracy. So for that, we tested under various number of populations and also the size. And we found no clear advantage we found in our case with lateral pan data. So the inclusion of the random terms often lead to the model non-convergence issue, resulting in using a single climate variables in the case the lattice and also the one did for the oak in Europe. And using a single climate variables sometimes can be problematic. For lateral pan, if we use the mean annual temperature for both provenance test sites, we can have a very high R-square. However, when you look at the spatial pattern, you can see that it's misleading. The productive area is not in the real location. And after we add more climate variables, then we got reasonable spatial distribution patterns of the productivity or height growth. So the trade-off between including random terms and limiting the number of climate variables in the universal transfer function is a concern. So, and the factors limiting the climate niche model to predict the fundamental niche model is that the occurrence data is affected by interspecific computations, the physical barrier, and the binary output. So in order to predict the fundamental niche model, we use a URF, which can overcome all this because the common garden experiment doesn't have a interspecies computation. We can overcome the physical barrier. We can use the probability as output. So we predict the fundamental niche at a global scale for lateral pan. And it matches well with most of the observations where they have a successful introductions. So I'm running out of time. Okay, let me quickly go to the landscape genomic approach. You know, the genomic information is rapidly accumulating. So, however, the genotype-phenotype association is remain a challenge. And although the genomic prediction has some promising results, and anyway, here I'm showing results from landscape genomics to use for seed zone delineation. So it's essential to have the delineation for seed and breeding zone is essential for seed sourcing and the breeding programs. For large pine, we have different number of seed zones from different studies. And the principle of the seed zoning zonation is that we need to minimize the within zone variation, and while we still have a reasonable number of zones to manage. So it has been done based on common garden experiment, which are, you know, expensive, long-term, and not sufficient for many, or even not available for many tree species. So there's also ever-lasting debate how many seed zones we should keep. So we used the gradient forest model with a large number of snippets from adaptation project. We use the climate variable scale-free climate data from Climatic, and we can define the the species spatial genomic variation for the species in BC and Alberta. Then, based on the spatial genomic variation, we split it into different number of zones. We look at the change of within zone genomic variation is reducing with the number of zone increasing. But we don't see a clear turning point. So then we also calculate the reduction of the within zone variation. We found that here is a clear cut. So we determined that 6 zone is the most logical number of zones. So we compare our zones with the earlier studies. We found that 65% agreement with Lippi's study, but we found it shows some advantage because the Lippi's method has clear impact from the political boundaries, and ours are independent of that. We also compare with another zone based on the genetic correlations. And they have 4 zones, we have 6, but we found we can separate the clear difference between the interior and the coastal climate. Okay, in summary, the genetic climatic mismatch at the species and the population level is the key of the most problems caused by climate change. There are many problems, but this is one of the key issues. Nature has created many species and genetic lines for different climate conditions. It's just a matter how we use them in a way. Climate niche models provide effective tools for climate matching at the species level, and the transfer and the response function can be used to help the seed sourcing and the seed orchard planning at the population level. And the fast accumulating genomic information has a great potential to serve as an alternative to our conventional approach. Here is my group. Everybody is working on climate matching at different levels from different aspects. Here are our sponsors, and thanks a lot. Sorry for the overtiming. Any questions, please?
Thank you very much for an excellent talk, Tongli, and for taking the time to join us from Canada. I'm sure there'll be some questions, so I'll just open the floor. Would anybody like to kick off? Catherine.
Thank you, Tongli, for this nice talk. I have a question about how we can learn from a comparison of climate niche modeling of the different species.
Okay, how to compare climate niche modeling across different species?
Not only comparing, but what kind of information we can learn from these comparisons of modeling between species, which could be helpful to complete the view for each of the different species. Could we consider that comparison between species could be more informative than just regarding climate niche modeling from one species independently?
Sorry, there are some part I didn't get it, but let me repeat your question. How can we learn from modeling different species to see? There are something I didn't get.
Yes, it's just could we learn from modeling in different species to improve modeling for one species?
Oh, I see. I never thought about that. We model many species. Each species has their own features. And some species is easily to model. It has a reasonable result. Some species is different. And you need to really balance the absence, the proportion of absence than the present data. It's hard to generalize. But anyway, we are working on modeling as many species as possible, then put them together. And traditionally, we are looking at from the species point of view. But I think it's important from the planting site point of view. When you have a planting site, you model all the species, and then you can make selections which species is the best for the given test size. I'm not sure I'm completely answer your question.
Okay, yes, I think that did. Thank you very much. Any other questions? If I could maybe ask one.
Sure.
I just wonder, do you think the use of the universal response function in overcoming the limitations of the numbers of test sites, not for a lot of species, might open up the potential to make use of a lot more sort of historic experiments that have been laid out that maybe don't have the geographic scale of some of the experiments you presented?
It can overcome to some extent, but still is the number of test sites need to cover at least a quite a range of the climate conditions. You know, URF use both provenance climate and the site climate. If so they can refer each other to strengthen the model, but this can help to some extent, but not if you only have less than 10 test sites cover a quite narrow range, that is still problematic.
I have a question, a very naive one, coming a little bit from outside. I'm not a modeler, but have you tried, or are you thinking, or do you know about people doing models using more than one trait, multi-trait models, that in a way that you could predict responses, not for just height, or but for an integrated phenotype?
Yeah. Sorry, you know, due to the audio effect, I have problem to hear from the question from the audience. Could you repeat it? I mean the navigator. Yeah.
Yeah, I was asking. The models you have been showing in your talk are for one single trait. Like
yes.
And I was wondering if there is also possibilities to do models for more than one trait together, like multi-trait model combining different, to get to model a full phenotype.
Good question. Good point. We in our demo, the first studies, we model the tree height. And for the study in Austria, we also model the diameter or the basal area. And but we model them independently. We have never thought about combining different traits to make multivariate analysis for a universal response function. It can be complicated. I haven't thought about this to put them together. So far, my suggestion is to model them independently, put them together, or say you can model the volume instead of height and the diameter. The volume is an integrate trait. It's a good point. You can explore it.
You presented an interesting slide about the number of provenance trials needed to have an accurate model. And the curve was decreasing a lot, down to 40 sites, as far as I remember. So it means that if we want to install common garden or provenance trial dedicated to adaptation, if we have 40 sites covering various climates, this would be enough to set up accurate models.
So the model I show, the results show is for a number of populations. So it's a common, many provenance trials have several tens populations. For the number of test sites, yes, the limitation of test size is the issue. You know, the random effect seems when your number of population or size very small, it has a slightly better performance. But the problem is the non-convergence problem. When you have the random effect in the model, you cannot include more climate variables. So that's a challenge. We really try to, you know, between the transfer distance, using transfer distance and using the provenance climate, we prefer our URF over UTF. However, I thought that introducing the random effect is a good idea. We tried different ways, but the results is not encouraging.
Anyway, I guess that the random effect is would make the black box for the part of variation you can't explain. If you have enough sites to include more than one climate from the side, then it might be explained and not need anymore to this experiment thing. But I had another question. I was discussing this issue with Rosniak models. They say, why don't you first do a high growth function and then expand the factors? You know, regular or whatever, and then you get it 2 or 3 parameters of this function, then you expand it by the time factors. It would might be another way to make one adjustment for 10 years and one for 15 and one for 20, but to model it.
Sorry, I can barely hear your words. I don't know. The audio effect from audience is very poor. Could somebody repeat?
Ouais, je me demande si ça serait pas mieux dans le micro.
C'est quel le micro qui prend?
I can try to repeat the question. Can you hear me clearly?
Yes, it's still not clear, but yes. Let's confirm every point I get. The microphone in the front is good, but in the audience is so poor.
Let's try here. I was discussing this with growth and yield modelers, and they say, why don't you first adjust the growth model, a high growth model, and then you get the parameters of the function, and then you can expand the function by site or climate factors. Then maybe you would get these parameters to be compared between sites and provenances or populations. And that might be a smarter way to study the phenomenon high growth than to do, let's say, discrete age adjustments when you have the problem that not all provenance are measured at the same age and so on. So do you think that might be a feasible approach?
Sorry, you said you develop the parameters for a site and the provenance first, right?
Yeah.
How to do that?
By high growth adjustment, so age and height, and then you adjust a function for high growth.
Oh, you mean the age adjustment?
Yeah.
Oh, if you have if you have a provenance trials established at different years, so now you have different ages, how to combine them together, right?
Exactly.
Okay. Yes, we did a study in the Chinese Cyprus, and that's established different years. So we did the year as adjustment first, then we develop your universal response function.
Okay, thank you. I think we have one more question, if that's okay.
Yes, I have a question about the possibility to be this kind of model self-updating. Well, with the URF or UTF, we can predict the expected performance or value, but we also have the interval of confidence of the model. So my question, if you believe that this kind of results could be also used to decide where new common garden experiment should be planted, maybe on the limit of the prediction or where the prediction is more uncertain.
Yes, that's a good question. You know, as I shown that for large pan, we have 60 test sites across province. And when we plotted against the climate, we found 20 of them are concentrated at mean annual temperature of 3. So that's a lot of repetition. We need to determine the climate conditions. Now we have climate data available, right? To pre-select the test sites instead of at the 70s, year 70s, there's no climate data available such widely. So they decide the test sites based on geographical distributions. Now we can use the climate model to predict the climate conditions, then to determine where is the best place to put our test sites. and also the sampling of the populations. So the AMAT trials established recently, we need much smaller number of test sites and the populations.
Thank you. I think that sounds like a very good place to stop. I think that gives us good way forward for future work. And I'd just like to thank you again for taking the time to be with us and to answer a lot of questions. It was an excellent talk and I think a really good way to draw today to a close. So thank you very much.
Thank you very much. How to...
Okay, and I think that draws our session for the day to a close. So thanks everybody for all the contributions and good questions. I think it was a really interesting day. Look forward to more tomorrow.
Thanks.